Dear friends, in this video I will talk about the economic botany and heterosis breeding in cotton. As you all know, cotton is one of the most important fiber crop as well as commercially important crop of India as well as world. The botanical name of cotton is Gossypium species, belongs to family Malvaceae with chromosome number diploid cotton 2n equal to 2x equal to 26 and tetraploid cotton 2n equal to 4x equal to 52. It is the most important commercial crop which is soft, fluffy, stable fiber. It is regarded as king of apparel. India is traditional home of cotton and cotton textiles. It is the only country which cultivates hybrids on commercial scale. One of the important commercial fiber crop which is known as white gold or king of fibers. Though synthetic fibers have made inroads in many countries in the world, still cotton occupies the prime position in India, constituting more than 70% of the total fiber consumption in the textile sector. Let us see the origin and distribution of cotton. The origin is Central Africa. It is well distributed around the world, including America, Africa, India, China, Brazil, etc. History of cotton. The earliest evidence for cultivation of cotton was in Old World, some 7,000 years ago, that is 5th to 4th millennia BC, by the inhabitants of Indus Valley civilization. Arab merchants brought cotton clothes to Europe during 800 AD. By 1500, cotton was known generally throughout the world. Importance of cotton. It is most utilized fiber. It has variety of uses. It is must for day-to-day life next to salt. Clothing and household items are the largest uses, but industrial products account for the use of thousands of bales of cotton. Cottonseed oil is used in lubrication industry. Top 10 cotton producing states. First one is Gujarat, followed by Maharashtra, then Andhra Pradesh, Punjab and other states. Taxonomy of cotton. Belongs to kingdom Plantae, division Magnoliophyta, class Magnoliopsida, order Malvaceae, family Malvaceae, genus Gossypium. Species of cotton. Totally 45 diploid species in genus Gossypium, which includes 6 important allotetraploids. The genus is divided into 7 genomic groups A, B, C, D, E, F and G. Few important species of cotton are Gossypium hirsutum, barbedans, arboreum, herbaceum, darwini, sturtinium, thurberi, tomentosum and raymondi. Classification of different species of cotton. Old World cotton are herbaceum, arboreum, which belongs to Asiatic distribution, then Anamalum and Stocksy. These are having genomic class of C1E1 and chromosome number basic number 1313, and Anamalum is from Australia. It is distributed in Australia, and Stocksy in Indo-Arabia. American cotton, it consisting Gossypium thurberi. The genome is D1 with basic chromosome number of 13 and it is distributed in Northern America. Then Herkensi D2 13 chromosome number, then Gossypium aridum D4 and Raymondi is D5. D5, that is Raymondi, is distributed in Southern American part. Coming to New World cotton species, Gossypium hirsutum, it is AD1 tetraploid. The big chromosome number haploid chromosome set is 26, which is distributed in Northern America. Next is Hirsutum subspecies genomes ADDH that is also having haploid chromosome set of 26 and belong to North America. Then Gossypium barbadans AD3 having 26 haploid set and it also belongs to South America. Cultivated species of cotton: Gossypium arboreum and herbaceum are Asiatic diploids having chromosome number 26, and Gossypium hirsutum and barbadans are allotetraploids, which are most popular. Coming to floral biology of cotton, flower is axillary or terminal flower having 3 to 5 bracts. Apicalyx means whorls of fused bracteoles. Calyx is gamosepalous. Corolla is polypetalous. Androecium, in this stamens are numerous in number and monoadelphous. Then gynoecium, that is female part. Here carpels 5 syncarpous, ovary multilocular and superior. It has single style and club shaped stigma. Taxonomy: belongs to order Malvaceae, family Malvaceae and genus Gossypium. Chromosome number of diploid is 26 and tetraploid is 52. The scientific classification, same one. Cytogenetics of cotton, this has have already covered, like it is having 6 genomics group. Then B, C and F genomic groups found in India. C genome is found in Australia and D in America. I repeat, B, C and F genomic groups are found in India. Then CE belongs to Australia and D to America. Botanical description: cotton plants have very prominent erect stem. Leaves are arranged alternatively and spirally around the stems. Leaves are petiolated and lobed. The stem and leaves are pubescent. The large leaf veins bear external glands known as nectaries and excrete a sweetish fluid. This is a branch, this is main stem leaf, and the second one is subtending leaf. This is node between two nodes. The internode is shown here. The bases of two leaf are known as axil. Then this stalk is petiole and this is fruit branching, and this is square which is just appearing in the branch. Here cotton plant with leaves removed shows the straight growth habit of the main stem and vegetative branches. This is after removing the leaves. In the second picture, a fruiting branch with leaves removed shows its zigzag growth habit. That means where fruits are found, zigzag habit, and where leaves are found, straight habit of stems are found in cotton. Floral biology: it has floral morphology plus floral anthesis. Anatomy of mature flower: this structure just resembles the hibiscus flower. This is anatomy of bud. These are flowers, how the flowers change their color after few days. This is the white color flower during the initial days, and day 2 it changes its color to slight pinkish, then bright pinkish, and finally after day 5 or 7 it will start making the balls. Stages of cotton development: first flower development will take place, then ball development, and after that bur development, that will yield the final cotton. Cotton flower: this occurs singly or solitary, produced on sympodial branches from the terminal buds. Base of flower surrounded by 3 leaf like triangular bract, that is called as apicalyx. Calyx is reduced to a small cup-shaped structure with 5 lobes. The branching of cotton is of two types. One is vegetative or monopodial branching and second is fruiting or sympodial branching. Vegetative structurally like main stem and sympodial branches bear flowers and fruits. Anthesis, twisted corolla emerges from the bracts. Variation in time of flower opening, it starts from 8 am up to 10 am. Temperature also affects the flower opening. Stigma becomes receptive soon after flower opening. Stigma receptivity remains for 24 hours and pollen viability will be for 12 hours. Coming to pollination habit, self-pollinated crop basically, but this is divided or classified in often cross-pollinated crop because pollen is shed directly on stigma. Cross-pollination is due to insects that ranges from 5 to 25, sometimes exceeds 50%. Hence, cotton is often cross-pollinated. Coming to fertilization and fruit set, maximum if pollination is at the time of anthesis. Fertilization is completed in 36 to 40 hours after pollination. 6 to 8 weeks from first flower to ripe fruit. Fruit is a ball. Young fruit or fruit bud is called a square. Selfing techniques. Let us move to hybridization techniques. In that first one, selfing techniques. Here different special techniques are discussed. First one is paper bag method, then ring method, mud smear and other methods. This is paper bag method where the bud is covered with a mature bud is covered with paper bag in order to prevent the out contamination. Crossing techniques includes emasculation, pollen collection and artificial pollination. The first step is selection of flower bud. The crossing work is initiated after 1 week of flower initiation. The flower buds of proper stage, that is buds which are likely to open in the on the next day, are selected for emasculation. Such buds have generally cream color and are well developed. This is a bud ready to be selected. Emasculation and crossing technique, hand emasculation and crossing, then Humphrey and Tuller method. Emasculation. An incision is made at the bottom near the calyx, then a circular shallow cut is given so that corolla and stamen tube surrounding style come off. Bud bag with butter paper and tag it. Hand emasculation and crossing. It is done in female parents on the evening using nails of thumb of both the hands. The staminal column along with anthers and attached petals are removed. During next day morning, the anthers of male flowers are rubbed against the stigma of emasculated flower and then bagged. Pollen collection. Collect flowers from the male parent during early morning hours, then collect pollen in straw tube. Pollinate next morning between 8 am to 1 pm. Insert the straw tube containing pollen on emasculated flower stigma like this. Fertilization and fruit set. In hand emasculation, 65 to 70% success will be seen. After fertilization, white flowers turn to pink or red. Coming to breeding objectives of cotton, the primemost objective is high yield, that is more number of balls, bigger balls, high lint percentage or high oil percentage. Then early maturity, superior fiber quality in terms of fiber length, fiber strength and fiber fineness. Resistant to diseases and insects, that is abiotic and biotic stress, then photo and thermo insensitivity. Current priorities for breeding in cotton are germplasm has narrowed considerably. The cotton industry around the world is upon improving yield, fiber quality, and pest resistance. Requirement of indigenous and international market has also had its impact on the objectives aimed by the breeders. Selfing and crossing techniques, this I have already told. Crossing for this is also discussed, for which open bud by rotating petals in anti-clockwise direction. We have to remove the stigmatic portion. When the bud is slightly opened, insert a drinking straw pipe into the opening in such a way that stigma and style come inside the pipe. Cover the opening of the pipe by mud or sticky soil, then bagging is not needed. Pollination is done by dusting the desired pollen into the hollow tube of straw pipe and then again cover it with mud. This is mud smear technique. Coming to heterosis breeding, some of the scientists who contributed for heterosis breeding, first scientists are Mel and Balls. They reported concerning heterosis expression in F1, that is in the cross between Gossypium hirsutum into Gossypium barbadans. Kulkarni and Khadilkar in 1929 reported exploitable magnitude of heterosis in interspecific crosses. Shantaram in 1951 showed 158% increase in F1 over the local parent in diploid interspecific hybrid. Patel in 1971, he is a father of hybrid cotton technology in India. He demonstrated heterosis at commercial level. He developed the world's first interspecific hybrid in cotton H4, that is a cross between Gujarat 67 into American nectarless. In 1971, Katharki showed heterosis in interspecific tetraploid hybrid through the release of Varlakshmi, that is a cross between Lakshmi into SB289E. Srinivasan during 1972 have described the procedure of producing hybrid seeds using male sterile lines where female parent is genetically male sterile conditioned by 2 genes and hand emasculation is not required. Davis in 1978 has discussed the problem and prospects of hybrid cotton and suggested the use of honeybees in affecting cross pollination while utilizing 3 lines A, B and R hybrid breeding program. Singh in 1989 has discussed the problems and prospects of hybrid seed production using male sterility. Those are lack of simply inherited restorer gene that maintains fertility over a wide range of environments, lack of development of good combiners possessing male sterile cytoplasm and restorer factor, then lack of dependable and economic method of controlling pollination by insect pollen vectors. Prerequisites for heterosis breeding are magnitude of heterosis, percentage of outcrossing, floral biology and availability of male sterility. Genetic basis of heterosis. In this, Surser et al. In 1986 reported epistasis effect while involved dominance effect in cotton and higher heterosis in hirsutum for yield was due to overdominance effect for bole number and weight. Bhattade during 1984 observed that heterosis in inter and intraspecific diploid crosses for yield and ball number was mainly due to non-additive gene action. Second step is level of heterosis. Higher level of heterosis is observed in interspecific crosses than in intraspecific crosses. Heterosis is high for yield and ball number, moderate for ball size and seed oil content, low forgiving outcome, and various fiber properties. Factors affecting heterosis: geographical and genetic diversity, agronomic performance, adaptability, and genetic base. General features of hybrid vigor. Heterosis is observed for many characters in different combinations of Gossypium. F1 hybrids produce exceptionally high and uniform quality of fiber. Parental line G67, Lakshmi, SB289E, Bikaneri Larma, LRA 5166, etc. have contributed to the production of high heterotic sustainable hybrids. Very high range of fiber category requirements has been met by hybrids. The diploid degree of inbreeding depression is low and implies the potential of exploiting the F2 heterosis also. The extent of heterosis observed in interspecific hybrid was 10 to 138%. The intra-hirsutum hybrid heterosis was ranging from 7 to 50%. In both cases, under high favorable environments, 80 to 187 has been observed in India. Exploitation of heterosis. It depends on two main factors. First one is careful identification of good combiners, and second one is success of hybrid seed production. Combinations which are favorable to produce successful hybrids in cotton are: first one is Gossypium hirsutum into hirsutum, that is intra-hirsutum cross or hybrid. Second is hirsutum into barbedans, this is interspecific HB hybrid. Then herbaceum into arboreum, a cross between diploid into diploid, this is interspecific Asiatic hybrid. Then arboreum into arboreum, that is intra-arboreum hybrids. General features of hybrid vigor in cotton are: heterosis is observed for many characters in different combinations of Gossypium. Male sterility in tetraploids. Genetic male sterility. In this, total of 11 loci have been identified that condition GMS, and 10 of them are in Gossypium hirsutum and 1 in Gossypium barbedans. MS4, MS7, MS10, MS11 are dominant and produce complete sterility, while remaining 7 are recessive. Among recessive genes, MS1, MS2, MS3 cause male sterility when present singly, while MS5, 6, 7, 8 behave as duplicate recessive genes. GMS system involves MS5, MS5, MS6, MS6 found in Greg MS is the only stable source utilized in India, Pakistan and USA. All Gossypium hirsutum genotypes which carry MS5 or MS6 are restorers. Any Gossypium hirsutum line can be converted into GMS system by repeated backcrossing with alternate selfing and selection. CGMS. In this, interaction between nucleus of Gossypium hirsutum and cytoplasm of Gossypium harkensi, Gossypium oridium, Gossypium trilobum induce stable sterility. Male sterility in diploids. First one is Hisar source. The recessive GMS in Gossypium arboreum cotton variety DS5. GMS1 was isolated as a spontaneous mutation. Here, note down that DS5 is also called as GMS1. Semi-closed corolla is drawn back in GMS1 and it has been overcome in GMS2. Akola source. The GMS like GAK423A is developed by transferring the genome of Gossypium anamelum cytoplasm. Different pollination techniques are hand pollination and bee pollination. Chemically induced male sterility. In this, chemical male sterilizing agents act by prohibiting antherdisons, chemical FW450, that is sodium A dichloroisobutyrate, and TD1123 have been demonstrated to possess male gameticide action in cotton. Coming to BT cotton and colored cotton, Bacillus thuringiensis is gram-positive soil-dwelling bacteria. It is also occur naturally in the gut of caterpillar of various types of moths and butterfly, first discovered in 1901 by Japanese biologist Ishiwata. Upon sporulation, it forms crystals of proteinaceous insecticidal delta endotoxins called crystal proteins or cryproteins. Encoded by crygenes, crytoxins act against nematode lepidopterans and hymenopterans. Bt serves as important reservoir of crytoxins, crygenes for production of biological insecticides and insect-resistant male sterile system that is against some insects. When insects ingest toxin crystals, the alkaline pH of their digestive tract causes the toxin to become activated. It becomes inserted into insect's gut, causes cell lysis and eventually kills the insect. To talk more about BT cotton, now it has covered widely in India, and it is the only commercial crop or non-food crop which is widely accepted for commercial cultivation. Coming to colored cotton, J.M. Vreeland reported several lint colors like brown, pink, blue, green, khaki, red in all the cultivated 4 species. Lint color determined by group of genes located at 3 loci, that is LC1, LC2, LC3. By the combination of these 3 loci, different colors will be formed. They are dominant over white alleles and operate in association with modifier genes that are suppressors or intensifiers. In the presence of strong suppressors, white lint is produced. Often, genes for lint color are pleiotropic. They control more than one trait. This has been the most important problem in development of economically and technically superior colored cotton. Color development in the fiber occurs between 30 and 40 days after ball formation. Solvent extraction experiments have shown that pigment may belong to the flavonoid group. First commercial success came in 1988 when Sally Fox of Natural Colors USA succeeded after 8 years. This is colored cotton, and these are the cloths prepared out of khaki colored cotton. Various color cotton developed by US Dharwad. Color hybrids produced at CICR by crossing natural color strains in Gossypium hirsutum with white strain. Coming to different hybrids developed by different cross combinations. First one, species involved. First one is intra-specific hybrid. The popular hybrids are H4, H6, H8, H10. Intra-hirsutum hybrids: JKHY1, JKHY2, PKVHY2, NHH44, Savita, Surya, Fateh, LHH144, Dhan Lakshmi, Maru Vikas, Om Shankar, DHH11. Then intra-arboreum hybrids are LDH11, AAH1. Coming to interspecific hybrids, the popular interspecific hybrids are Varlakshmi and DCH32. Tetraploid hybrids are NHB12, HB224, DHB105, TCHB213, NHB302, and Shruti. Diploid hybrids are DH7, DH9, DDH2, and FA46. Coming to tetraploid hybrids, all intra-hirsutum and interspecific hybrids between hirsutum and barbadans. Then diploid hybrids are all intra-arboreum and interspecific hybrid between arboreum into herbaceum. Methods of hybrid seed production in that conventional and sterility based. Conventional hybrids are all the four species are involved in production of hybrids, and then male sterility means popular are Suguna, PKV, HY3 hybrid 3 or PKV hybrid 4, MEC4, AAH1. Male sterility in tetraploid. In genetic male sterility, this has already discussed in the previous slides. Coming to popular hybrids released in 2002, these are BT cotton hybrids. MEC 12 BT by Mexico. Then in the same year, MEC 162 BT, MEC 184 BT released by Mexico, which changed the scenario of BT cotton widespread in India. Then in 2004, RCH 2 BT by Rasi company. Then in 2005, 16 hybrids by different companies, and recently Punjab Agriculture University developed PAU BT series hybrids, which became very popular. The list of different cotton hybrids which are suitable for different agroclimatic regions as well as different states are listed in these slides. And their specific characters are also given here. Gossypium hirsutum into barbadans gave rise to Varalakshmi during 1972. It was improved for fiber length. Then DCH32, DHB105, TCHB213, HB244 were for fiber fineness and strength. Then herbaceum into arboreum, the hybrids developed were DDH2, DH7, DH9, all were for high yield and drought tolerance. Coming to hybrids released from different research centers, the first center is PAU Ludhiana, which released Fateh LHH44, LDH1, and recently the BT cotton varieties. Then HAU Hisar released Dhanlakshmi and AAH1. Then Shri Ganganagar RAU, they released Maruvikas. Then Sirsa released Om Shankar, Surat Hybrid 10. Then Khandwa JNKVV, they released JK Hybrid 2. Then Akola released PKV Hybrid 3 and PKV Hybrid 4. Guntur released LAHH4, then Dharwad released DHH11, DHB105, DDH2, then Coimbatore released Surya and Shruti. Thank you. If you liked this video, kindly subscribe my channel. Thanks once again.